For millennia, the story of human health has been told through fragments. We had ancient texts describing terrible sicknesses and skeletal remains showing signs of trauma. But the true culprits, the microscopic germs and viruses that plagued our ancestors, remained largely invisible. But now, a remarkable scientific breakthrough has allowed us to hear the whispers of these ancient infections directly from the past. Scientists, led by a team of evolutionary geneticists, have embarked on an unprecedented journey through time. They have carefully collected and sequenced the DNA from the bones and teeth of over 1,300 individuals who lived and died across the vast expanse of Eurasia, creating a continuous timeline of disease that stretches back an astonishing 37,000 years. This groundbreaking work, published in the journal Nature just this month, is like opening a time capsule of human suffering and survival. The method itself is a marvel of modern science. Within the mineral matrix of bone and the hard enamel of teeth, tiny fragments of genetic material can survive for tens of thousands of years. This material doesn't just belong to the human who was buried. It also belongs to the pathogens that were circulating in their bloodstream at the time of their death. By isolating and piecing together these minuscule fragments of ancient pathogen DNA, researchers can identify the specific germs that infected people long ago, giving us a direct window into the diseases that shaped our history. The sheer scale of this study is what makes it so powerful. From the hunter-gatherers of the last ice age to the farmers of the first settlements and the warriors of the Bronze Age, the skeletons tell a universal story. The researchers were able to identify the genetic signatures of 214 different types of pathogens, painting a vivid picture of the microbial world our ancestors inhabited. What they discovered was that illness was not a rare or occasional event. It was a constant and ever-present feature of human existence. In fact, the genetic calling cards of various infections were found in approximately 75% of all the skeletons they analyzed, a testament to the relentless pressure that disease has placed upon our species from the very beginning. Before this research, our understanding of prehistoric disease was based on inference and educated guesses. We could see evidence of poor nutrition or bone deformities, but we could not name the specific bacterium causing an infection or track the spread of a virus across a continent. Now we have a map, a genetic atlas of ancient diseases. We can see which microbes were common companions and which were devastating invaders. This new knowledge transforms our view of the past, revealing that the great dramas of human history, migrations, the invention of farming, the rise of cities, were all profoundly influenced by the silent, invisible world of germs. If we travel back in time to a world before farms and cities, we find small, mobile groups of hunter-gatherers. Their lives were intimately connected to the natural world, moving with the seasons and the herds of animals they hunted. The new genetic study reveals that these ancient people were far from free of disease. The oldest individual analyzed, a person who lived some 37,000 years ago, carried a familiar set of microbes. The DNA found within their remains did not belong to exotic, terrifying plagues, but rather to a range of common oral bacteria. These are the very same types of germs that can cause gum disease and tooth decay in people today, a sign that some of our microbial companions have been with us for a very, very long time. For tens of thousands of years, this appears to have been the dominant pattern. The diseases that afflicted hunter-gatherer populations were likely those that could survive in small, isolated groups of people. These would be media group, macro shot of bacteria cultures in petri dish, animation of bacteria lingering in human body, environmental bacteria in nature, at stock, distribute _ equally chronic infections that could linger in a host for a long time, or common environmental bacteria. The genetic record from this long era shows a remarkable consistency. The primary infectious agents identified are the ones that live in our mouths and on our skin. This makes a great deal of sense, as the lifestyle of these early humans did not encourage the rapid spread of epidemic diseases that we are familiar with from later history. Living in small nomadic bands had a profound effect on the types of diseases people encountered. A fast-spreading illness like measles, for example, requires a large interconnected population to sustain itself. It would quickly burn through a small hunter-gatherer group, leaving no one left to infect, and then die out. Therefore, the germs that thrived were the ones that could be passed from mother to child, or those that could exist quietly within a person's body for years. The discovery of these persistent oral bacteria suggests a long-term, stable relationship between humans and their microbes, a kind of microbial baseline that existed for much of our species' history. It is important to understand that while these infections were common, they were not harmless. Chronic gum disease can lead to tooth loss, which would have been a serious problem for people who needed strong teeth to process a tough diet. It could also lead to systemic inflammation, weakening the body's ability to fight off other threats. So, while our earliest ancestors were not facing down waves of plague, they were in a constant low-level battle with the microbes they carried with them every day. Life was a struggle for survival, and the ever-present threat of infection was an inescapable part of that struggle, a quiet hum of danger beneath the surface of daily life. Everything changed when our ancestors began to farm. This shift, known as the Neolithic Revolution, began around 10,000 years ago and represents one of the most significant turning points in human history. People stopped wandering and began to settle in one place, building permanent villages and eventually bustling towns. They cleared forests to plant crops like wheat and barley, and they began to live in much closer proximity to one another than ever before. This new settled way of life created the perfect storm for the emergence and spread of new infectious diseases. The genetic record uncovered by scientists shows this change with startling clarity, marking the beginning of a new and deadlier chapter in our relationship with germs. The study reveals that a significant change in the types of pathogens infecting humans appears consistently in the genetic record starting around 6,500 years ago. This is when we first see the rise of blood-borne pathogens and zoonotic diseases, those that jump from animals to people. Living in larger, denser communities meant that an infection could spread from person to person with terrifying speed. Furthermore, the storage of grain attracted rodents like rats and mice, which carried their own array of dangerous microbes. Sanitation was often poor, with human and animal waste contaminating the water and soil, creating a breeding ground for bacteria that could cause devastating intestinal diseases. Perhaps the most crucial change was our new relationship with animals. Humans began domesticating sheep, goats, cattle, and pigs, bringing them into their settlements and living side by side with them. While these animals provided a stable source of food, milk, and labor, they also carried a host of pathogens that were new to the human body. The research provides direct genetic proof of this crossover. Shortly after these animals were domesticated, pathogens like relatives of cowpox, the bacteria that cause leptospirosis, spread through animal urine, and meat-borne yersiniosis began to appear in the DNA of our ancestors. Our new farmyard friends had unwittingly opened a Pandora's box of disease. This transition fundamentally altered the landscape of human health forever. While hunter-gatherers contended with chronic, persistent infections, early farmers faced the new threat of explosive epidemics. The very innovations that allowed human populations to grow, agriculture and settlement, also made them profoundly more vulnerable to disease. This period marks the beginning of a biological arms race between humans and a whole new suite of pathogens. The benefits of a stable food supply came at a terrible cost, a cost paid in sickness and death as our immune systems struggled to adapt to the dangers of a world we had created for ourselves. Among all the diseases that have haunted humanity, few cast a longer or darker shadow than the plague. Caused by the bacterium Yersinia pestis, it is infamous for the Black Death, the pandemic that swept through Europe in the 14th century, killing a vast portion of the population. Historians and scientists have long sought to understand the origins of this terrifying disease. It was widely believed that the first plague pandemics occurred during the Bronze Age, around 3,000 to 4,000 years ago. However, the new genetic evidence from this comprehensive study has forced a dramatic revision of this timeline. It reveals that the plague is far older than we ever imagined. The researchers made a startling discovery in the remains of an individual unearthed in Western Russia. Within the DNA extracted from this ancient person, they found the unmistakable genetic signature of Yersinia pestis. The skeleton was dated to be 5,500 years old, This single finding pushes back the known history of the plague by a staggering 2,000 years. It means that humans were falling victim to this deadly bacterium during the late Neolithic period, long before the great civilizations of the Bronze Age had even begun to rise. This ancient strain of plague was an early version of the killer we know from the history books, a ghost from a much deeper past. The discovery is significant not just because it changes a date in a textbook, it tells us something profound about how the plague evolved. When scientists analyzed the genome of this ancient 5,500-year-old strain, they found it was missing a key gene known as YM. This particular gene is crucial for the bacterium's ability to survive inside the gut of a flea. This suggests that these very early forms of the plague were likely not transmitted by flea bites, the method that allowed later strains to spread so rapidly and cause such devastating pandemics. Instead, this early plague probably spread through direct contact with infected individuals or animals, a slower and less efficient mode of transmission. This finding provides a crucial piece of the evolutionary puzzle. It shows us a snapshot of the plague in the process of becoming the super pathogen of later history. It had the potential to cause severe illness, but it had not yet acquired the genetic tools needed to fuel a continent-spanning pandemic. The emergence of plague 5,500 years ago coincides with a period of increasing human settlement and trade, creating new opportunities for pathogens to spread. The discovery of this ancient victim is a chilling reminder that the deadliest diseases often have long, hidden histories, evolving in the shadows for millennia before they emerge to change the course of human history. Section 5. Great migrations and great plagues, the second wave of infection. You see, the invention of agriculture, well, it triggered the first major shift in human disease, but it certainly wasn't the last. A new study has identified a second dramatic surge of infections that began around 5,000 years ago. And this wave, interestingly enough, wasn't driven by people settling down, but rather by people on the move. It was linked to the mass migration of pastoralists from the Eurasian steppe, a vast region of grassland stretching from Eastern Europe to Central Asia. These groups, often associated with the Yamnaya culture, were master horsemen and herders. They moved across huge distances in horse-drawn wagons, and as they traveled, they brought a deadly cargo of new diseases with them. These steppe migrants, well, they weren't just carrying their culture and their languages, they were carrying the germs that had evolved alongside them and their domesticated animals. The genetic evidence shows that as these groups spread west into Europe and east into Asia, they introduced a cocktail of dangerous pathogens to local populations that had never encountered them before. The study found that this is when the plague, which had existed in an early form, began to spread more widely, but it wasn't alone. The migrants also brought with them the bacteria that cause relapsing fever and the parasite responsible for vivax malaria, a debilitating disease that would go on to plague humanity for thousands of years. For the local Neolithic farming communities that had been living in Europe for centuries, the arrival of these migrants and their diseases would have been catastrophic. These local peoples would have had no prior immunity to these new pathogens. Imagine a sickness sweeping through a village where no one's immune system has ever seen it before. The result would have been devastating waves of death, a biological shock that would have destabilized societies and cleared the way for the newcomers to settle. This evidence suggests that the great migrations of the Bronze Age were not just a clash of cultures, but a clash of microbes, with deadly consequences for the losers. This finding adds a crucial new layer to our understanding of prehistory. It shows that human mobility is a powerful engine for the spread of disease. The horse and the wagon, celebrated as innovations that connected the world, also created the first prehistoric pandemics. The study allows us to see this process unfold at a genetic level, tracking the movement of pathogens alongside the movement of people. It is a stark illustration of a principle that still holds true today. When previously separate populations connect, the risk of new and dangerous epidemics soars. The Great Migrations of 5,000 years ago were a grim preview of what was to come in an increasingly interconnected world. When we think about being sick, we often imagine fighting off a single illness. But the reality, both today and in the ancient past, can be far more complicated. One of the most fascinating and honestly grim discoveries from this extensive study is the clear evidence of co-infections, cases where a single individual was infected with multiple, distinct pathogens at the same time. This finding adds a new dimension of horror to the experience of disease in the ancient world. Facing one serious illness is bad enough. Battling two or more at once would have dramatically increased the chances of a fatal outcome, weakening the body on multiple fronts and overwhelming its defenses. A particularly striking example of this phenomenon was found in the skull of an individual from the Viking Age. When researchers sequenced the genetic material preserved within it, they were astonished to find DNA from two very different and very serious diseases. The person was infected with the hepatitis B virus, a pathogen that attacks the liver and can cause chronic illness. At the same time, they were also infected with Yersinia pestis, the bacterium that causes the plague. It is a truly terrifying combination. An immune system already struggling to fight off a persistent viral infection would have been severely compromised, making it far more vulnerable to the fast-acting and deadly assault of the plague. This evidence of co-infection helps us understand why mortality rates during historical epidemics might have been so high. It's likely that many people were not perfectly healthy when a new plague arrived. They may have been suffering from other chronic or low-level infections, malnutrition, or parasitic worms. These underlying conditions would have acted as a powerful accomplice to the newly introduced pathogen. The study suggests that co-infections were not a rare curiosity but a common feature of life. When one disease weakens the body's defenses, it opens the door for other opportunistic infections to take hold, creating a vicious cycle of sickness. Understanding co-infections in the past has important implications for how we view health today. In many parts of the world, people still face the burden of multiple simultaneous infections, such as HIV and tuberculosis, or malaria and bacterial pneumonia. This ancient DNA evidence provides a deep historical context for this modern problem. It reminds us that an individual's overall health is a complex picture, and that the impact of any single disease can be magnified by the presence of others. The Viking who died with both hepatitis B and plague tells a personal story of immense suffering, but also a broader scientific story about the compounding dangers of disease. Our DNA is not just a blueprint for building a body, it is also a history book. It carries the genetic scars of the battles our ancestors fought and won against disease. This vast study of ancient pathogens provides one half of the story, the germs. By comparing it with the evolution of the human genome over the same period, we can see the other half, how our bodies adapted to survive. The research offers direct evidence of natural selection in action, showing how the rise of new diseases drove powerful changes in our own genetic makeup, changes that continue to affect our health and well-being to this day. A clear example of this evolutionary pressure can be seen after the agricultural revolution. As people began living in larger settlements and with domesticated animals, they were exposed to a barrage of new pathogens. This created an intense selective pressure. Individuals with genetic variants that helped them survive these new infections were more likely to live long enough to have children and pass on those helpful genes. The study found that after farming became widespread, certain human genes, known as alleles, linked to inflammatory responses became much more common in the population. These genes essentially put the immune system on high alert, making it better at fighting off new bacterial and viral invaders. This adaptation was a crucial advantage in a world rife with infection. A more aggressive, inflammatory response could mean the difference between life and death when faced with a dangerous new bacterium. Those with a quieter immune system were weeded out, while those with a more trigger-happy one survived. Over thousands of years, these protective genes spread throughout the population. We are the descendants of these survivors. The genetic legacy of our ancestors' struggle is written into our own DNA, a testament to their resilience in the face of relentless microbial assault. Our immune systems are in part a product of the Neolithic farm. However, these ancient genetic adaptations come with a modern-day trade-off. The very same genes that helped our farming ancestors fight off infections are now associated with a higher risk of autoimmune disorders in the 21st century. In our modern, highly sanitized environments, our immune systems do not face the same constant barrage of pathogens. An immune system that is genetically primed for a major fight can become restless. With no external enemy to attack, it can sometimes turn on the body itself, leading to conditions like Crohn's disease, rheumatoid arthritis, or multiple sclerosis. The ghost of our ancient evolutionary past haunts our modern health, a powerful reminder that adaptations that are beneficial in one environment can become a liability in another. The study of ancient diseases is not merely an academic exercise to satisfy our curiosity about the past. It holds incredibly valuable lessons that have direct applications for doctors, scientists, and public health officials working today. By resurrecting the genomes of ancient pathogens, we can watch evolution in action over millennia. This provides a unique roadmap of how germs change over time, helping us to anticipate their future moves and develop more effective strategies to combat them. The secrets held within 37,000 years of bone and teeth can help us protect the health of billions of people living now. One of the most promising applications is in the development of new vaccines and treatments. Pathogens are constantly mutating to evade our immune systems and our medicines. This is a huge challenge for vaccine designers who must target a part of the virus or bacterium that is stable and unlikely to change. By comparing the genomes of ancient and modern strains of a pathogen, scientists can identify which parts of its genetic code have remained conserved or unchanged over thousands of years. These stable regions are likely essential for the pathogen's survival and make excellent targets for new vaccines, as the germ will find it much harder to evolve resistance against them. The ancient plague genomes provide a perfect example. The discovery that the earliest strains lack the YMT gene, which is critical for flea-borne transmission in modern pandemic strains, reveals a key evolutionary step. It pinpoints a specific adaptation that turned a dangerous disease into a global catastrophe. Understanding these pivotal moments in a pathogen's history can help us identify the kinds of mutations that could make a current animal disease more dangerous to humans. This knowledge can inform surveillance efforts, allowing us to monitor animal viruses for the specific genetic changes that might signal an impending jump to the human population. Furthermore, this vast database of ancient infections does not exist in a vacuum. Researchers can now combine this genetic information with other large datasets, such as ancient climate records and models of prehistoric land use. By doing so, they can build sophisticated models to understand the environmental and social conditions that increase the risk of zoonotic spillover events. This helps us answer critical questions. How did changes in temperature affect the spread of malaria? How did deforestation and the expansion of farmland bring humans into contact with new animal diseases? This historical perspective is vital for predicting future disease hotspots in a world undergoing rapid climate and environmental change. The story written in the DNA of our ancestors is ultimately a story of survival. It is a tale of a relentless 37,000-year-long struggle against an invisible world of pathogens. This epic narrative, uncovered from the dust of graves, reveals that illness has been one of the most powerful forces shaping human history and our very biology. From the persistent oral bacteria of Ice Age hunters to the devastating plagues that swept through the first farming communities and traveled with migrating peoples, our species has been constantly challenged, adapted, and transformed by disease. We are the descendants of those who endured. This research dispels any romantic notion of a disease-free natural past. It shows us that every major innovation in human history came with new microbial risks. The warmth of the first settled communities also fostered the spread of infection. The domestication of animals, which provided food and labor, also introduced a host of deadly new germs. The invention of the wheel and the wagon, which connected distant cultures, also created highways for pandemics. This deep history teaches us a humbling but vital lesson. Human progress and disease have always been intertwined, two sides of the same coin. One of the most profound insights is how this ancient struggle continues to affect us today. The immune system genes that protected our ancestors from Neolithic pathogens now leave some of us vulnerable to autoimmune diseases. This is a powerful reminder that we are not separate from our past. We carry it within ourselves. This knowledge must guide our approach to modern medicine, encouraging a more personalized view of health that takes an individual's deep evolutionary history into account. It also highlights the hidden costs that can accompany even the most positive societal changes. Ultimately, looking back into this deep history provides us with a guide for the future. It underscores the ever-present threat of zoonotic diseases, reminding us that the majority of new infections will likely come from the animal kingdom, a process that began thousands of years ago. By understanding the evolutionary pathways of past pandemics, we are better equipped to forecast, prepare for, and fight the pandemics of tomorrow. The voices of the long dead speaking to us through the language of DNA are not just telling us stories of how they lived and died. They are giving us the wisdom we need to survive.